All right, welcome to part 2. When we left off, we were talking about the endomembrane system, and I had mentioned that when proteins are first formed, they are going to be, if they're going through the endomembrane system, they're going to be created in the rough ER. They're going to move through the rough ER and then be transported via a vesicle to the Golgi. And as they move to the Golgi, remember the Golgi is the place where proteins get their final modifications, and they get ultimately targeted for delivery to which part of the cell they need to go. So there are 3 possible destinations of a newly synthesized protein that goes through the endomembrane system. It can either go to the exterior of the cell, to the plasma membrane, either to become part of the plasma membrane or to be released to the external area of the cell. It can go to a storage, a vacuole, which we'll talk about in a second, or it can go to a lysosome, which is a structure that can help destroy and remove proteins that are misfolded or folded improperly. So to look at the lysosome is basically just another membrane-bound sac. It is like a vesicle, but this vesicle contains digestive enzymes. And so when you have another vesicle that contains things that you want to digest, a lysosome will fuse directly with that vesicle. And so now you have a bigger vesicle full of digestive enzymes. So in this example, you're seeing food that's being brought into the cell. That food is in a vesicle that then fuses with a lysosome, and the lysosome can then digest the food that is found inside of that vesicle, breaking that macromolecules down into simpler molecules so that the cell can utilize it for energy. The other way you might see a vesicle and a lysosome being used is for removing proteins or organelles that are no longer useful to the cell. In this case, it's going to be a process called autophagy. So now you form a vesicle around an organelle you no longer want. So this cell no longer wants something like this mitochondria or this peroxisome. So it forms a vesicle around them, fuses that vesicle with the lysosome, and then everything inside will become digested. So you can see it's the exact same process. It's just that the cell can either bring its lysosomes to bear on external things coming in, such as food, or internal things that the cell just wants to get rid of. Now, the vacuole is another location that materials can end up in. And so the classic example of a vacuole is what we call the central vacuole, which is found in a plant cell. And this is a giant sac of water and nutrients and other chemicals for the cell. It's storage for a later time. And so that gives us 2 of the locations that proteins might end up as they go through the endomembrane system. And of course, the third place they might end up is the plasma membrane. And so to look at the whole process from start to finish, we can begin with DNA that's found in the nucleus. The nucleus contains that DNA, but it does allow RNA, mRNA, to exit through the nuclear pores. And that mRNA can find its way to the rough ER, which is studded with ribosomes. Those ribosomes can turn that mRNA translated into protein. So that protein will then travel through the endoplasmic reticulum, and as it travels through, it's getting folded and attachments and chemically modified until it reaches the end of the ER, and then it moves through a vesicle to the Golgi. Again, it's going to process from the cis side of the Golgi to the trans side of the Golgi, and as it does, more modifications are being made. And eventually, when it reaches the trans face, it can be pushed out in a vesicle to destroy all the protein if the cell didn't do it right, and then it would associate with the lysosome to digest it. And as I mentioned, it can go from a vesicle to the plasma membrane, and everything inside of this vesicle can be released to the outside world. Or this vesicle could end up going to, which is not pictured, a vacuole, which would just store those proteins for a later time. All right. The one thing the endomembrane system doesn't mention, or I haven't mentioned, is that the proteins don't have to be made on the rough ER. There are also free-floating ribosomes in the cytoplasm. Those can also translate proteins, but if you make a protein that's not part of the endoplasmic reticulum or the endomembrane system, it's going to be destined to be inside of the cytoplasm of the cell. If you want to get it outside of the plasma membrane or in the plasma membrane itself, it has to process through the endomembrane system. All right, so next set of organelles we want to talk about are the mitochondria and the chloroplasts. And again, the mitochondria and the chloroplasts are going to be covered in much more depth later in the year, but for now I want you to know that these are places where we change energy from one form to another. So mitochondria do cellular respiration. That's the thing that animal cells and plant cells do to use oxygen to generate ATP. So you break down sugar using oxygen and you create ATP, which is a simple form of energy the cell can use. The flip organelle of that, the opposite organelle of that, is the chloroplast. That's found in plants and algae, not in animal cells. This is where photosynthesis occurs. Photosynthesis is going to use sunlight and carbon dioxide to generate sugars, which can then in turn be broken down by the mitochondria, which is why a plant cell would have both of these structures. Lumping in with mitochondria and chloroplasts, we're also going to include the peroxisome, which is an oxidative organelle. It helps reduce reactive oxygen inside the cell so that it is less damaging to the cell. So the interesting thing about the mitochondria and the chloroplast is that both of them are found inside of a double membrane. And so the prevailing theory about how these came to be is that the mitochondria and the chloroplast were once simple prokaryotic cells that were engulfed by a eukaryotic cell and created a symbiotic relationship where the prokaryotic cell was benefiting by being inside of a larger protective eukaryotic cell. The eukaryotic cell benefited by being able to utilize that energy that the prokaryotic cell inside of it was generating. And so you ended up with a relationship that moved forward through time to the point that now all eukaryotic cells have a mitochondria, and many eukaryotic cells, those that are plant cells, contain a chloroplast. Other evidence for this endosymbiont theory that I'm speaking of, along with the double membrane around these structures, is that the mitochondria and the chloroplast actually have their own genomes. And that's a relic of the fact that they were independent organisms, and they are able to replicate themselves using that genome. And in fact, we can do some research with lineage by looking at mitochondrial DNA. All of you in this room will have the identical mitochondrial DNA to that mitochondrial DNA of your mother, your grandmother, your grandmother's mother, your grandmother's mother's mother, all the way back in your maternal lineage. And we'll talk later about why that is, but that's an interesting feature of the mitochondria, that it is sort of a separate entity inside of your cells. So for now, the only real things you need to know are sort of the structure of these organelles and kind of know what they would look like. One thing to know about the mitochondria is it has a lot of membrane. So you increase the surface area by having these crista moving in and out, and the membrane ends up being the site where ATP is formed, which we'll cover in much more depth later in the year. So the more membrane you have, the more ATP you can make. And you see this sort of structure that has all of the crista is inside of another membrane. So you've got that double membrane that makes up what we call the mitochondria. Similarly, if you look inside of the chloroplast, you also have a lot of membrane. You have what we call thylakoid, these tiny membrane sacs. They stack up into granum, and so you've got a bunch of stacks of membrane structures inside of the chloroplast. Again, the reactions that make the chloroplast work occur on the membrane, and so the more membrane you have, the more activity you're going to get out of the chloroplast. So just be able to recognize these structures and know generally that the mitochondria makes energy from sugar, and the chloroplast makes sugar from the sun's energy. Now, the peroxisome, I don't have a lot for you to look at. We have just a TEM image here. But the peroxisome's function is to reduce reactive oxygen. So hydrogen peroxide would be an example of that. It's going to turn that peroxide into water, and then it's just a normal thing that the cell can dispose of or utilize for other purposes. It's just a way to clean up sort of all of the reactions that are occurring inside of the cell. So the last concept I want to talk about is the cytoskeleton. So the cytoskeleton is structures that are inside of the cell, a network of fibers that actually organizes what's going on and holds everything together. You often think about a cell as being just a giant bag of liquid, but in fact it has a large number of fibers that connect everything, organizes everything that's going on inside, and hold everything together. And you can see this in this fluorescent image, and we've looked at fluorescent images before, but the green in this image would be the microtubules, and the red would be the actin or the microfilaments. So these are two different types of cytoskeleton. You can see that they're pretty ubiquitous throughout the cell itself. So one of the things these fibers can do is they can allow things to move along them. So they can take something from one side of the cell to the other side of the cell, basically by using another protein that's able to walk along an element of the cytoskeleton, like a rope or a highway. So in this case, we have a vesicle. This would be a membrane sac containing stuff. It would attach to a protein that's able to walk along, and it literally walks along the fiber like I'm doing with my fingers here. And as it walks along, it drags that vesicle behind it, and so you're able to move things quickly throughout the entire cell. So this figure kind of highlights the three cytoskeletal fibers you need to know. The microtubules, which are the largest, and they make hollow tubes. The microfilaments, which are the smallest, and they make these little twisted fibers. Microfilaments I often will just call actin. And then the intermediate filaments are sort of in between the size of the microfilaments and the microtubules, and they are another sort of cabled protein here. All of them have sort of similar functions. They maintain cell shape, they help with cell motility, and they move things around inside of the cell. And there's your microtubules again, microtubules here in green. Here are your microfilaments in red, and your intermediate filaments in this picture are in green. You see the intermediate filaments are kind of all over the place. Actin tends to be closer to the edges, and microtubules tend to radiate out from one location inside of the cell. So that location that the microfilaments actually radiate from is called the centrosome. The centrosome is a structure that's composed of two centrioles. The centrioles are also made of microtubules, and you can think of the centrosome as just an anchor onto which all of the microtubules can attach. So if you want to pull against microtubules and move things throughout the cell, you have to anchor it somewhere so that you're actually able to move against the microtubule and not pull the microtubule away from where it's supposed to be. So one of the things you should know about microtubules, one of the features that a microtubule can do, is it helps form the eukaryotic flagellum. This is a different structure than the prokaryotic flagellum because it actually contains a large number of proteins and fibers and microtubules. I'll give a little shout out to this protein right here, which is the one I did all of my thesis work on. But the way this works is that you have a large number of microtubules. They're linked together with motor proteins between them. And when they are linked together, they can't slide past one another. But so when the motor proteins actually walk, they push against until the flagella bends in one direction, and then it bends in the other direction. You do that over and over again, and you get that nice beating motion that we tend to think of in a flagella. So again, this is just an example of how a fiber can be used for motility in a cell. Fibers can also be used, actin can be used to help a cell walk along, so it can at the edge of the cell form a pseudopodia, a fake foot, and reach out and kind of pull against itself to help the cell move. So that is all I want to talk to you guys about cells. So concept 4.7 is about extracellular components and connections between cells to coordinate activities. I'm going to save that for the next unit, chapter 5, when we talk about the membrane, because I think it'll make a little more sense to you when you understand the membrane a little better. So importantly for you, 4.7 is not going to be on your chapter 4 test. So for your chapter 4 test, I need you guys to review 4.1 through 4.6, review the material that we did in class, and make sure that you can go through all of the sections of the study outline except for 4.7, and you have a good idea of what's going on in a cell. I hope these video lectures helped, and please feel free to email me if you have questions. I'll be happy to help straighten out your understanding.